Sometimes, in science, we're trying to study one thing and end up accidentally discovering something else. Cancer researcher Dr. Cathy Wilson from the University of Cambridge recently experienced such good fortune. While trying to understand the function of cancer gene MYC in mice, a gene that goes haywire in almost all human cancers, she accidentally ended up achieving the holy grail of heart disease research, making heart cells start dividing to make more cells. I'm Eva Higginbotham, and I spoke to Dr. Wilson about the happy accident.
So we had this model where we switched on and off MYC, and we looked at what happens. And as we kind of expected, when we switched on MYC in tumor-prone organs like the liver and the lung, we saw cells start dividing, start proliferating. But in tissues like the heart and the brain, we didn't see any proliferation. And this is not unexpected, but we were a bit confused why. If we were expressing MYC in the same way and it should be functioning in the same way, why wasn't it having an effect in these normally non-regenerative organs? And we looked at other different proteins that are expressed in the heart, and what we found is that there was a protein complex called PTEF-B, and this is called positive transcription elongation factor, and this seemed to be really lowly expressed in the heart. So we thought perhaps MYC was really dependent on the level of this protein complex. So what we did is we overexpressed half of this complex, cyclin T1, in the heart together with MYC, and lo and behold, we were able to make these hearts start proliferating.
Wow, so you're literally making the cells make more cells in the heart.
Exactly.
The heart normally doesn't regenerate so well. So normally heart cells don't divide and make new cells, even when there's an injury because of something like a heart attack. Why is that?
So the heart doesn't regenerate after an injury, and no one really knows why the cardiomyocytes can't start proliferating, can't dividing. And like in other organs in the liver, if you damage the liver, it can virtually replace half of itself via proliferation and growth. So if we could harness this MYC and cyclin T1 to regenerate damaged hearts, that would be the most amazing end goal.
And that would be sort of like a real cure for the results of a heart attack, right? Rather than mostly it's sort of current therapies for after you've had a heart attack are more about managing the scar tissue that forms. Is that right?
Exactly. Well, there's therapy where you can limit the death of cardiomyocytes. So after a myocardial infarction, billions of cardiomyocytes die within the first few hours. So if you can limit the death, then that is one potential therapy. But yes, it's trying to limit the scarring. But if we could just regenerate those dead cardiomyocytes in that first short term after the myocardial infarction damage and death, before any scarring has happened, then this might be a window of opportunity for us. So there's so many hurdles to navigate, to jump over. Can we put in MYC and cyclin T into people's or animals' hearts, and does it cause a functional benefit once we've damaged a heart? Yeah, how do we get these genes into the heart? So while maybe gene therapies try to alter the genetic code, I guess everybody's heard a lot about CRISPR-R therapy and actually modifying the DNA, but we wouldn't want to do that, because if we were going to modify any MYC DNA, it might cause cancer. So we're really looking at RNA therapy. So traditionally, RNA could be injected into the cell, but it actually caused an innate immune response and cells ultimately died. But now there's a number of drug companies developing modified RNA technology. So this is where a modified nucleotide is put into these RNAs, and this changes the secondary structure so they don't get recognized by the immune system. And this is perfect for our kind of delivery system of MYC and cyclin T1. It would lead to a short-term, localized, intense burst of expression of protein with no genetic footprint. And so this is really important when using cancer genes. So if we could inject these into the hearts of people that have just had a myocardial infarction to reactivate a bit of cardiomyocyte proliferation to regenerate the heart just after damage, that would be the most amazing thing.